Hi, everyone. My name is Bhamidipati, and I'm going into my third year as a biology major here at Drexel. I'm on the pre-med track, and one of the topics I'm very interested in is neuroscience, which is actually a big part of the reason I decided to do this project. For those of you who may not know, Huntington's disease is a progressive neurodegenerative disease, meaning that it causes the progressive loss of neurons in the brain. One of the ways in which this disease actually affects the brain is by hindering dopamine transmission between neurons. And this can actually impair attention and cognitive function. This disease affects hundreds of thousands of people around the world, and current treatments and therapies have only been able to manage symptoms. There is no present cure for Huntington's, but previous experiments have shown that a drug called SK609 improved sustained attention deficits in rats by binding selectively to a specific dopamine receptor, the dopamine 3 receptor, without desensitizing it. Therefore, this study tested an analog of SK609, or the C2 drug, and its ability to rescue locomotor and longevity defects associated with the Drosophila model of Huntington's disease. The way this study was completed was by crossing male Drosophila with the mutated HTT gene with female Drosophila virgins with the ELAP gene, resulting in the Huntington's disease model Drosophila flies in the progeny. A longevity assay was performed which tracked the survival of the Huntington's model flies when given the C2 drug food. And this data was then compared to the survival of wild-type flies and Huntington's model flies that were given regular food. A locomotion assay also tested the abilities of Huntington's model Drosophila larvae, which were given C2 drug food, and their ability to move across grid lines on an agar plate within 30 seconds. And these results were again compared to the positive and negative controls. The results of the study suggested that the C2 drug is actually capable of rescuing longevity defects in the Huntington's disease model flies to a similar level as the wild type, or healthy flies. Our preliminary data for the locomotion assay similarly suggests that the C2 drug rescues locomotive function for a Huntington's disease model Drosophila larvae, but a larger data set needs to be tested before this conclusion can be made.